Hi everyone, welcome to our 21st multi-omics reading group. And today we have Haotian Cui for this talk. So I would introduce the speaker for a little bit. Haotian is a researcher specializing in machine learning, genomics, and drug discovery. He recently completed his PhD in computer science at the University of Toronto, advised by Professor Bo Wang. His research focuses on developing large-scale self-supervised and generative foundation models for single-cell omics and molecular biology, integrating AI with experimental pipelines to accelerate therapeutic discovery. He's the lead author of scGPT, which is very famous, one of the first generative models for single-cell multi-omics. He also led the development of LUMI Lab, autonomous AI-driven platform for mRNA therapeutics. He has published numerous papers in leading journals and conferences, including Nature, Nature Methods, Nature Communications, ACL, EMLP, SML, etc. So today, thanks Haotian for accepting the invitation, and now it's all yours.
Thank you so much for the warm introduction. Let me start by sharing the screen. And it's a pleasure to report here today, and talk will be around 30 minutes. Feel free to interrupt anytime in between for questions. It's not very easy to use Google Meet. For example, I cannot really see the discussion in chat. So if anything happened and a question there, maybe Xinyu, would you let me know if there's any question pop up. So first, let me do a very quick summarization of our scGPT, and also a very quick intro for the single-cell foundation models for people who are not very familiar with this direction. This will be followed by my personal takes on several lessons learned after the release of our work. First, what are single-cell omics and why we care about building a foundation model for it? Different from bulk-level readout, single-cell sequencing provides cellular scale resolution and reviews cell heterogeneities. For example, this can be conducted by droplet-based single-cell RNA-seq, where individual cells are barcoded so their RNA risk can be reviewed, separated in later stage. Recent advancements enable multimodal sequencing across the central dogma. For example, ATAC-seq can be used to measure the chromatin accessibility, and SAT-seq measure RNA expression and protein altogether. CRISPR-based perturbations also have been combined with RNA sequencing in the perturb-seq, so the effects of genetic perturbations can be recorded in high throughput. This reservoir of technologies can provide comprehensive views of individual cell states. Today, we will focus on technologies that mostly read cellular RNAs. For example, single-cell RNA-seq. It comes in large abundance. You may find around 100 million of varying cells in public domains. And the technology around RNA sequencing is also more mature, providing higher data quality. These features make it suitable training data for a foundation model to answer relevant biological questions. Now, once we have a foundation model, or even a virtual cell model, if you may call it, we can use it to answer many, if not all, questions in the field. From more biological-related questions like cell state assignment, gene regulation predictions, to more therapeutic-related questions, such as predicting perturbation response, differentiation trajectories. So more importantly, if we think about it, these application tasks are interconnected. For example, as visualized here, solving gene interactions may greatly contribute to the solution of other questions, such as how cells differentiate. So this kind of a relation justifies the rationale of foundation models, and it can learn from and also provide a very large shared biological context that is useful for many applications, and they can share the context together. First, so when we really care about building the things all together, we care about the feasibility. While texts are made of words, cells can be characterized by genes, and this is an analogy that inspired us to believe transformers will be suitable to learn interactions and between gene tokens. And that's one of the first questions we first try to answer, is that what kind of model architecture you can use. Transformer is also very suitable here. And the second pillar we wanted to, we think that makes direction feasible is about the data. As we previously mentioned, we already have a scale of sequencing data, especially considering single-cell RNA-seq, such as shown in this screenshot from the cell-by-gene census, which contain tens of millions of sequence cells. And actually, this screenshot on the right was actually taken from 2023. And the public data corpus really grows in an accelerating speed over the last years, from 33 million cells from 2023 to around 108 million last time I checked in 225. So therefore, one can train a large model via self-supervised learning to build a pre-trained model that can understand cell states. We, in our which is one of the first work as a attempt to building this model, in scGPT, we conducted an autoregressive generative training. In the pre-training, we had large-scale expression matrices, and they are collected and pre-processed. We selected around 33 million healthy human cells from 51 organs and tissues. And the model differs slightly from Euro transformer you see and in natural language domain in terms of token embedding and also the transformer model itself. For the input embedding, we use 3 sets of tokens. The interesting one, I just want to briefly mention the interesting one, which is about this expression value. So the other things are more or less like tokens, like genes. You can imagine gene names can be the tokens, which is like tokens of words. And now we have, instead of tokens of English words or language words, now we have tokens of gene names. An interesting one is about expression value. You have to have a way to modeling the continuous expression counts or the expression magnitude of the cell states. So for this, we introduce a pre-processed way that map all the expression in quantized values, and then use a neural network embedding to then map the quantized values into dense embeddings. So when you combine the 3 embedding together, now the layers of embeddings together, now you can go to the core transformer layers. The core model is a 12-stack transformer block in scGPT with customized mask attention for generative training. Briefly speaking, the model is trained to reconstruct unthin gene expression, and a mask attention guaranteed only relevant information has attention weights for genes to be predicted. Now, the output of the model HL, you can see here, is a group of vectors with a length equaling number of genes. We also reserved a special token called the CLS embedding at the beginning of the output. So the output at this position, the CLS position, they can represent the integrated information, which in other words is a cell embedding. Now, scGPT supports varying applications after pre-training. These tasks are achieved either by fine-tuning or zero-shot calling of the model. So the model can do data integration, cell atlas querying, or cell type assignment, or sometimes you can also find the model off-the-shelf model, or a model after fine-tuning can predict genes that are related to certain cell pathways or certain enrichment in your perturbational data. And so the model can do varying tasks. We also build this is one thing I want to highlight. We build a fast reference mapping with the cell type embedding for mapping cells to very large cell atlases. Since we can store the atlas embeddings, you can, as I mentioned, the model can output a cell embedding, and you can call the model on very large cell atlas over 30, 40 million cells. You just need to call the model once and then store all the embeddings. And the next time you can include new cells, like your new query cells, very efficiently, again, by the model to have your query cell embeddings. And now you can call a nearest search algorithm, very fast one. Inference, for us, we implement that by the Facebook face. And then you can do this workflow to use it as a reference mapping tool that can find the nearest neighbor of your new cells from out of the very large cell atlas. So this can be conducted very fast, within seconds for thousands of cells. If you're having a GPU, that can be conducted within 1 second. And works as it performs in this way, and it works as a very fast reference mapping tools. And in another direction, one particularly interesting task scGPT did was the perturbational prediction, the perturbation response prediction. Now, In this task, the model learns to predict unseen perturbation results from known expressions and known perturbational experiments. Imagine you can train on the model on the training set of several perturbations, and I'll ask the model to fill out the unknown ones. What will happen if you predict the cell in an unseen perturbation? The model is trained, for this task, the model is trained to predict the target expression after a perturbation and given the input cell in the control setting. Here we fine-tune it and test the model on 3 perturb-seq datasets with 1 gene or 2 gene perturbations. Our method outperforms the previous state-of-the-art methods such as gears, and also demonstrating the predictions highly correlated with the ground truth. Now we also tried a reverse perturbation task, which means the reverse direction as we showed in the previous slide. Now, given the result state, we ask the model to predict the source perturbation, like which of the genes are the driving genes that can cause the perturbation result. This was showcased on a rather small subset of the data of 7 test cases. On the left-hand side, we show the most successful cases where the true perturbation combination is retrieved within either top 1 or the top 2 guys. And overall, a very high hit rate is observed among all test cases when compared to other gears or baseline of calling the most differentially expressed genes. To summary, scGPT provides a general pre-training workflow for the single-cell data and supports varying tasks. We were very fortunate that the scGPT, because of a quite popular tool in the field, and it gives us a chance to communicate and learn lessons from our users. First, scGPT indeed can generate biological meaningful cell and gene embeddings. Here I show an example of a very large retina dataset collected by a user group. You can see the pre-trained embeddings align very well with their human annotations. And here, the embeddings for UMAP is done by calling on our scGPT embedding, but the color is shown by how well it matches with a human annotations. When they tested on this newly collected, you can see here, when they measure that, it achieves over 95% accuracy. So this is actually a very common use case, as we heard from other users. When they directly use a model off the shelves, they can achieve a very high accuracy when they do this kind of reference mapping or cell type calling. But we observe here is this high accuracy is usually achieved on this very coarse cell types, coarse cell labels. What I mean here is that what they are not happy, users are not happy, is when looking at this more subtle cell state and subtype of cells. This is what remains to be missing. For example, actually this very large blob of broad cells can contain over 100 annotated subtypes. The pre-trained model does little job distinguishing them apart, at least not at this very higher level. Now for this case, we actually collaborated with the user, and later on they annotated these cells very well with fine-tuning. However, I do want to recognize this as a missing piece that is surely, the pre-trained model, we surely expect it can make more sense to have a more, updated capability to do well on this task. We also observe initial success about perturbational prediction and driving gene identification. Showing on the right is a case of a collaborator's, this is one of our collaborators' fibroblast dataset. The model first generates in silico driving genes conditions by a method examining the gene attention score change. Then we in silico perturb the cells and project it on the UMAP. I think there's my mistake. This is a wrong figure here, but the model effectively does very successfully on this kind of task when it predict the perturbation effect given a in silico prediction of what kind of genes. Here showing an example from another paper, GeneFormer, but scGPT does a similar task where it can predict a subset of driving genes, and later on it can be verified in wet lab studies showing that these genes can be the driving genes to drive a certain direction of cell state change. Here is an example not from scGPT, but from a related paper like GeneFormer, where they show that the predicted genes verified very well and aligns with the direction of reprogramming the cell to achieve a more direction aligned with the shift towards a iPSC cell state. But what is missing here is that this kind of study, also the figure shown here is what we previously looked at, the perturbational response fine-tuning, is that we observed several initial success like that, but usually that answer is done on small subset of data after fine-tuning. And when you do the model off the shelf in pre-training, or you want to call the model in a zero-shot setting, I think we often saw much less successful rate. So what is missing here is a reliable perturbational and dynamic modeling right after pre-training. Also, what we found, another piece of question that is very interesting, that one may say that this kind of missing problems of the model, either the cell type capturing or the reliable perturbational predicting capability. Maybe you can scale up the model, and maybe that is a simple solution just by the scaling law. However, we found that is not a very easy case. A universal scaling law is not very clear for single-cell models. We started by this looking at the scGPT. When we originally reported the scaling law was a scaling effect in the original paper, we were quite happy at that time. Particularly regarding the data size, we found that the large pre-training data size shown here, when you enlarge the pre-training data set from just 30K cells to 33 million cells, you do observe a improved performance on downstream task. Shown here is a performance on the cell type assignment task. However, if you look at more carefully, and you can find that it actually reaches a plateau after around 3 million cells, which is only 10% of the training data. And honestly speaking, after the release of the our paper and also the release of many following papers, we found this plateau or saturated phenomena is actually very common for single-cell foundation models. You don't really observe a scaling law after the data reach a certain size. On the other direction, when you want to scaling up the model sizes, we also found mixed results were reported. From the literature, also from our own experience. I particularly want to highlight this recent discussion, not for single-cell models, but for a related model. This is a figure showing for ProGen 3, a protein language model. You can see that the so on this this is a figure screenshot from their paper from the ProGen 3. You can see after enlarging the model size to a certain degree, for example, after around 3 billion models, the model's performance is also saturated, measured by the Spearman correlation on their validation set. This is actually a fitness score of the model to some variation test. I think this is for some gene mutation task. And but I want to show this because I found this very interesting hint of a key factor, which is a task alignment. This is a hint to explain why we have this kind of saturation for single-cell models or for other domains like protein models as well, is that when you're actually looking at the pre-training of the model, it scales very well. The validation loss keeps changing, keeps decreasing after the model reaches a 46 billion parameter size. So it tells us that the decreasing of your validation loss doesn't really give you a better model when you're evaluating the model in your actual applications. This is a phenomena here shown in the PLMs, but I believe there's also a similar phenomenon we observed recently in single-cell foundation models as well. So that is a hint for the solution of the work is to have more aligned training task. Hopefully we can find a task when you train your model in this way, you will also use a model in this way when you actually inference a model in certain applications. I will have more discussion on this in later slides. So with its reflections in my mind, we next discuss how the ongoing trend of the multimodal and multitask pre-training may address alignment challenges, as I mentioned in the previous slide, and perhaps bring about the future, in the future, bring about a better models for us.
Sorry, do you mind if we bother you for a few questions?
Not at all. Please go ahead.
So maybe we could have a little like interim kind of discussion.
Yeah.
I had a question about, I remember when scGPT came out, I remember reading the preprint, and kind of one of the key features I took away from it was the more like the causal next token prediction modeling that you proposed with scGPT that of course parallels GPT models.
Mm-hmm.
And that came in contrast with the more like the mass language modelings from like scBERT, for example. So do you think that using causal modeling for next gene prediction, essentially, does that help with the scaling properties you think for these single cell models? You think there's a benefit of that in terms of the scaling that you were just now discussing with us?
Yeah. In terms of whether using a causal model, whether it scales better, I think my answer is no. I don't think the solution to the scaling law is depending on your model architecture. I currently more believe it is as I mentioned before, the real key solution is we find a better alignment between the training task and your actual application. If you think about it, it's quite differs in our domain, considering comparing with natural language processing. When you have GPT-4, I mean the OpenAI GPT-4 that is trained to predict the next token, the next word, and is used to predict the next word as well. But that is not the case for us. I will mention that in later slide. I do think maybe perturbational prediction is a very good task. When you train the model to do perturbational prediction, actually it's shown on this figure. I call it dynamics modeling, to predict maybe the next state of the cells, either perturbation or maybe the natural development stage in the future. So if you train this way, and we can imagine many of the tasks are also set up in this way, so the training and the application can align better. That may be the key solution to scaling, I think. Yeah.
Okay, so the alignment of the task is more important, you think?
I think so, yeah.
Okay, thank you.
So I would like to chime in a bit on that. One thing is the alignment of the task, but something else I think about is this slide where you show how there's different segmentations at different levels in cell types, right? Because by nature, cell states are continuous, right? And then they get discretized up to some point. So after contrasting that with the slight scaling that is seen from the jump from a few thousand examples all the way up to 3 million, I couldn't help but think that the additional blur, or rather the lack of more information being extracted from these discrete spaces, comes from the fact that you will plateau after you're able to segment those cells in those basic groups, let's say, like the ones you showed in earlier slides. But then the problem actually comes from where you're trying to go through these more concrete states, or rather more dynamical states. Would you say that task alignment somehow has to do with the model not being able to discern between the finer states as well?
Oh, yeah. I need to rephrase or retract me a little bit on that. So I'm not saying the model doesn't have the capability to differentiate more fine-tuned cell state, because when we fine-tune the model, at least we can see the model can be easily fine-tuned in a certain way to reach a very good capability in differentiating the cell state. And also, I think you're referring to maybe, let me go back, something like this right here.
That's
so
exactly right.
you're referring to the model looks like only give you a very large cluster of the row cells, but really not differentiating it into subclusters. That is the case for this, but that is not the case for some other cell types, like the RPE, and also for what's this one, dark color AC. I'm not sure which one that is. I think a lot of the reason is because this is a very challenging data set, because in our pre-training, we didn't really have a very large retina data set in the pre-training. When we look at a cell types that are more aligned with our pre-training, maybe the immune cells, we do see the model has more capability in differentiating subcell types. So one thing is that I do believe the foundation model is one of the good way. I mean, a self-pre-trained foundation model is a promising way to really differentiate even fine-grained cell state with certain training. The advantage here is that actually, because we're training it by self-supervised learning, we do this unbiased training without the feedback of human annotations. So the model, during the training, the model doesn't really know whether it's saying a cell type as a core cell type or fine-grained cell type. It doesn't matter. It's just self-supervised learning. So a lot of the unbiased signal can happen there. But that is a part, the first part of the answer. The second part is that I think to really improve the to really validate the improvement of the model, the reason you see here, on the other hand, is because evaluation of a model is by looking at the cell type calling. Now, this is a not very good choice because a cell type assignment, a cell type calling, usually when you evaluate, you have to look at human annotations. And now you face the bias of human annotation. A lot of the annotations are based on PCA, based on linear separations of cells, and different group can have different measures. So those are the challenges. Even you reach a very high score, you can be biased to artifacts of seeing the improvements of the model, but actually that is not the case. Yeah.
So there I would be inclined to say that one sort of precedes the other in the sense that if you will be linearly, or rather just dimensionality reducing your space somehow, or dimensionally reducing your space somehow, and you're learning from those features directly, there is only so much you can actually bake into the model to make up for the loss of resolution, let's say. So it's a problem that has to be tackled at both that initial reduction level and maybe baking in additional constraints to the model later. That is, of course, just speculative, just a hunch. But if you were to focus on either of these, be it having a more informative initial space or baking in additional constraints to the model, what do you think would be the solution to the current pressing problems of the self-supervised foundation model space?
If I understand your question, can you elaborate more on the loss of the resolution? I think you mentioned that, the loss of maybe the cell state resolution.
Yes, of course. So LLMs do this sort of aggressive compression, right, when they learn by self-supervised paradigms, training paradigms in general. Mm-hmm. There is no way you're keeping the fidelity of raw RNA transcription space. So this is somehow, you have to tokenize, you have to compress somehow for the model to learn, and then you compress further. And that results in a certain loss of granularity. Do you think that a model will learn better if you could supply just the original data ambient space to your model as is with no additional constraints and have that somehow be coupled with a good training procedure? Or do you think that just having a reduced tokenized space is fine? That's good enough. So as long as you bake, let's say, very precise constraints or model training objectives.
Thank you. That's a very great question. I do believe in your first option you mentioned.
I see.
I think during the pre-training, we are trying to do it in a more unperturbed, unbiased way where the model can find the compression strategy itself that try to keep enough signal while reducing enough noise, right? Keep enough signal to fulfilling the self-supervised task. A lot of the compression, because a lot of the compression strategy is related to the task, eventually related to the task. Whether you're doing cell type assignment or you wanted to predict the cell fate, cell state change of the cell, that would require different compression or different dimension reduction of your tokens. So we don't really want to do that very early in the stage. Right? I think during the initial pre-training, you want to keep a holistic model that compress the information in a more general way, trying to keep enough information as you can. Maybe it lost certain kind of skill in your downstream tasks, but that can be later on fine-tuned or maybe in test time compute to improve the model once again in a later stage. Yeah.
Very insightful. Thank you.
Thank you. Yeah, I think this is where we left, right? Yeah. So after release of SGPT, Informer, and UCE, we see a lot of new progress in the last year. They can be largely categorized, into 5 directions. Here I listed here. A diverse view. We can see a diverse view. The field in the field appears. For example, many new training objectives can be added to the pre-training or fine-tuning stages, and they can be used to optimize different foundation model into different applications. One of the examples is collaborators and I also worked on some of these directions. For example, we recently released the SGPT spatial and also a MesoGPT for epigenomics modeling. Just to give a very quick overview of our SGPT spatial, it is an extension of SGPT onto the spatial omics. We collected around 13 million data points. Out of the public domain, we just curated 30 million data points that spanning different spatial omics technologies, and we use that to continually pre-train the model. So again, it can be used to address many applications of spatial data analysis, such as annotating the interesting regions in your spatial slide, and also impute unseen genes or certain cell types due to the limited resolution of your spatial profiling. We can use the technology to do super resolution on your spatial slide. Now, although the diversity of this progress, it does make one wonder, what is the most relevant next step towards virtual cells, building a more capable models. And my personal answer is about this direction. I call it dynamics modeling, focusing on perturbational training, mostly focused on the perturbational training. This is related to what we already discussed, and also the point I made about the alignment. The reasons for this are threefold. First, dynamics modeling is, is important, exemplified by, the perturbational prediction is closely related. This is a figure showing what you can use by modeling the perturbational prediction way. You can use a model from an original state to predict what kind of genes can guide the cell to a target cell state. You can imagine this kind of formula or workflow is very close related to therapeutic discovery, so it's very important for these important applications. And on another view, dynamics modeling, many of the training currently on single-cell RNA-seq data, we can think about its one dimension, static dimension about cell profiling. But the dynamics dimension is really another dimension. You can imagine if we train this way, we open a new dimension, a orthogonal dimension of more application potentials. Now, the second reason I think this is important is about the feasibility. Sorry, I think this is doable, is about the feasibility. We do have a large volume of genome scale data, genome scale perturb-seq data, or at least perturbational data publicly available. And for example, I want to mention the recently released Tahoe 100M, and also that was one data set. And also another data set released a couple of days ago is the Zera-Auring data set, which consists of millions of genome-wide perturb-seq data to cell lines. And that means training the data, especially in the pre-training stage, training the data in a perturbational way is more feasible now. Another thing, the third pillar of the reason is about, perturbational training or measuring the model's capability in predicting the future state of cells is actually an unbiased evaluation, meaning that unbiased from human annotation, So that is a more trustworthy way to measure the real progress. I think we briefly discussed in our discussion, all the kind of questions or limitation about human annotation. One to mention is here, a lot of the annotation are done on linear models or calling on marker genes, which is quite linear. Quite linear in the separation space, and also only usually only measures a certain cell state to a certain level of cell types change, but not to a very continuous change of the cell states. So because of the three ideas I just mentioned, I think it's doable and also important for this direction. the answer to a next way thinking about how we actually build it. In our recent perspective paper in published in Nature, we discuss the possible architectures we can use or possible algorithm we can use to fulfill this direction. Actually, that is a more straightforward discussion about algorithm. So the perturbational training can actually happen in a very natural way, fitting in the current training of the foundation models, fitting the current architecture of transformer models. First of all, a lot of perturbational data may come from multi-omics with perturbation as some of the new perturbations even coming in the spatial domain. But as we already show in our existing works, Azure has a separate model or has one model that is capable to modeling all the different approaches in a multi-scale way, with maybe different scale of your training tokens, of your modality tokens. And next interesting thing about dealing with multi-omics data or multi-task data is that you can have customized attentions dealing with different layer of your tokens. One can imagine certain things like a hybrid transformer layers with either intermodal attentions or intramodal attentions, where your attention can happen to predict the change within your one layer of omics or between different omics layers. A more interesting view related to the transformer architecture, also the pre-training way, is that we can really learn a lot of lessons from the larger language model in the natural language domain. We can the specific lesson I want to mention is that we can unify the tasks, a lot of the tasks, in this unified way of prompting the model in a sequence-to-sequence architecture. We can have the model with different prompt tokens, but the same architecture, and then use the same architecture to fulfill different pre-training tasks. For example, the thing I listed here is that we already have the single-cell RNA-seq model on the statics, training on the statics profile of single-cell RNA sequencing. Can you imagine the model is trained on looking on certain, here the blue tokens are, for example, a sequence token for your mRNA sequence or your DNA sequence somehow, and then you can use a model to predict the missing tokens in the same technology. But you can use the same kind of framework on many other tasks, like temporal prediction, cross-model generation. More specifically about the thing we talk about, we can use a model to predict perturbation training just by changing the test tokens. For example, here is an example of you can input the knockout information of which gene has been knocked out by your CRISPR introduced CRISPR system, and then use a model to, given your control cells expression, predict the post-perturbational effect of your cell state. Now, given this unified way, I think it makes it feasible to really have very large foundation models trained in hybrid, unified way across task and across data set. And it has the potential to have a model that can really model and understand all the cell state in your different contexts. Now, this is actually a more straightforward answer to fulfill this is a more straightforward direction and discussion about model architecture. I think the more challenging discussion about building the next state, the future generation of dynamics modeling, the real challenge really lie on the data collection. For this, I want to talk about the model-guided data collection, which I think could be even more important need for building of dynamics models. So currently, we have a lot of static single-cell RNA profiling, but we still lack of very large, high quality, and especially perturbational data. We do have perturbational data in cell lines, but we have less perturbational data in more related biological contexts like in vivo data. We lack this kind of data. And to illustrate this idea about model-based data collection, let me introduce a recent work of us in a related, while different field. We built a model-based self-driving system for drug delivery. And so the image shown here is our, we call it our self-driving lab version 1. We system, they are built to, this is our LumiLab system. They are built to make LNPs, lipid nanoparticles, for the drug delivery of mRNAs. So they contain all consecutive steps from synthesizing the lipid nanoparticle, delivering to cells, and also test in vitro and readout and feedback to our in-house chemical foundation models. This is really a closed loop that on the front end, you can see the robotics here generate the data. And on the back end, the very large chemical foundation model we build can propose a new RMPs, and that will be tested again in the next round. We achieve the throughput of 100 new RMPs per day per machine by using the LumiLab system. Here's 1 minute demo for the system. So the LumiLab you just saw, you really see the robotics working in the action. It contains 3 stages of training. It contains the core of it is a model for the chemical molecules. The model contains 3 stages of training. Stage 1 is a generic training on 30 million molecules, followed by a stage 2. Because we're making lipid nanoparticles, we have another 15 million lipid-like molecules. And stage 3 is what you just saw, the active learning with self-driving lab, where you have the model be fine-tuned in loops of accumulated data in the active learning loop. What we want to highlight here is that we do saw the effectiveness of stage 1 and 2. So the pre-trained model and also the continual pre-trained model with the lipid molecules shows much better correlated, a much higher correlation score on a pilot run we're showing up here comparing to other methods. The successful of the model is, here is just to give a context of what we were really doing and what we use the model in the active learning loop. For this application context, the model's task is to predict the potential lipids with a potential lipid structure that can be conducted in the next round of experiment. That comes from a very large search space of around 220,000 of lipids by this constructed by this multi-component chemistry library. And the model is used to predict, which one from this very huge library that is more promising in the next round, and is from an active learning perspective, where which one of the lipid can give you a much more information gain in your next round of model learning. And then we use a model prediction, and we use a model proposal and feed that into the next round of robotics generation. The 4 workflow of the LumiLab are as follows. We do have this active loop of 10 iterations while balancing the exploration and exploitation in each round of the experiment implementation. meaning that in each of the iteration, we use the robotics to run 2 plates of proposed lipids. One of the plate is on more promising lipids and give you a better result, but the other plate is on more unknown or uncertain lipids and can give you a high information gain. And here we show the constant increasing performance among iterations. And third, we found that actually the successful story of LumiLab is that we found this novel bromination strategy. The top 6 in vitro of the model's proposals are this kind of lipids that have this very novel bromine atoms showing up here in the lipid tails, which is a new strategy in the field. We later tested it as in vitro and in vivo. It achieves a better result consisting to the head-to-head comparison without the bromine lipids. And so this is a story of LumiLab that has been successfully applied, but in a slightly different domain. I just want to use that to, now we go back to the thinking about the single cell domain, that the LumiLab result provide the empirical conclusion that actually, the LumiLab within 10 iterations of search in a very large chemical space successfully found and give you a better family of lipid nanoparticles. We use that to showing that the guided exploring of the data collection and a optimization bimodal can give you a very accelerated search in a very large space, which is the same thing we need for the search for the perturbative data. I just want to mention a recent work called EvolvePro from the group at MIT also shows similar results with a model-based directed evolution, a closed loop of experiments. And given these inspirations, I personally propose a workflow for the perturbational training. You can imagine the model and data, how they co-involve together in the following way. When doing an initialization, we can have the genome scale perturbation data, which we already have in the public domain. We can use that to train a base foundation model. And a model can be used to give you under certain specific context, give you a starting proposal of what kind of perturbations you want to test in your application. For example, I want to give an example here talking about if you are trying to enhance your CAR-T therapeutics and trying to find co-stimulators, what kind of genes, if you perturb, if you knock out, or if you overexpressed, can be a stimulator for a better CAR-T. Imagine you select a pool of 1,000 candidates, and now you can run the model in this kind of iterations similar to LumiLab, and which is about using the model predictions and feed that into your wet lab study, and then collecting your in vitro result and use that to fine-tune your model again, and now the model can propose the next round of iteration of data. Now, it was highlighting this important feature of this active loops. First, things, by running this, we're not running in the genome scale studies in the public domain. We're running in a much smaller scale. But they may go beyond the cell lines and ties down primary cells or organoids. So thinking about this, I'm actually proposing a outer loop that can generate the data. You can imagine these data are like the cherry on top. We have a base of data that you can collect it in very large studies, but on cell lines usually, which you already have in the public domain. But this cherry on top give you a more guided data in a more interesting context can happen in primary cells or organoids. And in another word, is actually accumulating the most valuable data that fulfilling the gap between your cell line data, although they are in large scale, but fulfilling the gap between cell line data and the data that is more interesting in the human context. And secondly, when running these batches of loops, specifically for multiple different applications, one is essentially accumulating the most valuable data for varying applications in varying directions, which can be used in a future pre-training as well. So this outer loop can be a more feasible approach in the short term. I think by this I mean that instead of just one model fits all, we are calling the instead of expecting we have a large foundation model that can give you all the answer in the short term, I think it's more meaningful to have a lot of foundation model, but co-involve with your data collection loops to having them more adapted for certain applications. But after the short term, by this co-involved loops, we're also accumulating the next iteration of more valuable data to pre-train your next generation of models. So to summarize, this talk, I highlight the lessons I learned from scGPT, the importance of training, and also the inference alignment, the importance of the alignment between the training and the inference tasks. And we also discussed the trends for building future virtual sales models, including the multimodal and multitask training. And the outer loop of active learning, personally believe is a way to enhance the data collection and training efficiency. Thank you all. And I'd like to express my deepest appreciation to all my collaborators for their contributions. And I'm willing to take any questions.
Wonderful. I would like to thank our speaker for the absolutely amazing presentation. And with that, as he mentioned, we will now open the floor for any additional questions from the audience. Please write your question in the chat or speak up now if you do have one.
Maybe I will ask one question. Like, what's your current next step? Like in the current moment, since you already finished the spatial omics and also you are working on the perturbation omics. So what's your next step?
Oh, I think it's basically a couple of slides. I personally also collaborators are actively trying to propose the next pre-training for incorporating perturbation data into the pre-training. Yeah.
Okay. Thanks.
Yeah, we are expecting to release a model in the next few months.
Yeah. Have you ever tried to speculate more, like, or study why like the scaling law doesn't work on a single cell? Like, instead of just hypothesizing like the task alignments issue. Like, is maybe more deeper study on this?
Honestly speaking, not. A lot of the discussion is still empirical, but we're not. It's not just a hypothesis. We do find some signals internally about the alignment after we're doing the perturbation training with the new evaluation. We found that might be a more promising way. Yeah, but it's still more empirical and more preliminary. Yeah.
Thanks. So that would be in the, like you mentioned, you release the model within the month or next 2 months. Like it would be in the paper, like as well for the scaling experiment you mentioned.
Yeah, I mean, that is the current project I'm working on. We do hope we can release that as soon as possible. Yeah.
Oh, I see. Thanks.
No problem.
Dylan, you had a question?
Yeah, I had a question about the, you mentioned how you were collaborating with other people on methyl GPT. So that was one example of more of a, not necessarily multi-omic model, but just a model that diverges from RNA-seq or even perturbation modeling. Do you think that other modalities like spatial transcriptomics or methylation or any other types of chemical modifiers would help in a perturbational modeling?
That is a great question. So I think the short answer is yes, but followed by the feasibility is really doing that, or the data quality may be more challenging. We do have in vivo perturb-seq, or even I forgot the name, but basically perturb-seq in spatial omics. We do have this kind of data. We do have multi-omics perturbing the cells in multi-omics ways. But a lot of these in vivo perturbs are either coming relatively lower data quality, or they are not genome scale, or the volume of them is limited. So incorporating them in pre-training, I think in the long run is definitely the direction to go. But currently, you face this kind of challenges. But one part of the modality is quite interesting, is about the cell painting. I think is about people from recursion like that a lot. You instead of doing RNA sequencing, unbiased sequencing of the cells, you can do the imaging-based profiling of the cell in a more focused panel. But that give you high-quality data and also high-volume data. I do imagine there's a much higher opportunity combining this kind of, cell painting data and single cell data together in a pre-training model. I think that can be very promising.
Yeah. Thanks for that answer. And my other question was about you mentioned and I have your paper here on multi-omic modeling. So, and you did mention it here about longitudinal modeling. So maybe in a way, perturbation modeling is kind of related to longitudinal modeling, but with clear perturbation. So in terms of longitudinal modeling, were you also thinking maybe in terms of development, or what kind of questions do you feel you could tackle there? How would those be related to perturbational modeling?
Thank you so much. That's a part we discussed in the nature perspective, but I didn't discuss here. So basically, you are talking. Sorry.
I guess you have it here as well.
I that's right. Highlighted here in this slide, right? So the temporal prediction, or you call it, we also call it longitudinal generation. I think it's actually close related to perturbation training, and also that's the thing I call it, dynamics modeling. So imagine you can think about they're related in a certain way. Like longitudinal generation, here we mentioned, you can imagine a cell is differentiating. Looking at a stem cell, you're trying to predict, what's the next stage, what's the fate of the current stem cell. Is differentiating to CD4 plus T cell or CD8 plus T cell. This is related, just by, it's my personal hypothesis, this is really related. It's like the stem cell in a human body is also perturbed in a natural way, right?
That's right.
Have some transcriptional regulations. You can think about that. That can be your perturbational signaling. Now that is highly correlated to the perturbational signaling you have externally by introducing a drug or a CRISPR knockout. What happened to the cell state, the mechanism might be connected. So I think training the model in this multitask way, each of the tasks are interconnected with each other. You have a higher chance to build a model with higher volume of data, right, from different aspects to have the model to really understand this question. Yeah.
And the fact that you kind of also agree with the very strict relevance between longitudinal modeling and as a natural kind of process to study perturbation modeling leads me to yet another follow-up about when you discuss about reverse perturbations. You mentioned that in a slide. So in line with what we were just discussing now about longitudinal modeling, could you view, let's say, as cell reprogramming using Yamanaka factors as a type of reverse perturbation? So you kind of reverse the process of development, right? So you go from differentiated cells to cells with higher stemness.
Yeah.
And you introduce transcription factors to do that. So they're almost like perturbation agents, but they're a bit more, I'll say, natural, although they tend to be done in experimental settings. So again, back to this parallel between longitudinal modeling and perturbational modeling, is there also a sort of parallel between reverse perturbational modeling and sort of reverse differentiation? Do you feel that parallel also holds in the reverse direction? I don't know if that made a whole lot of sense, but
I think that's a great question. And honestly, this is also the first time I think about your proposal, right? This connection between the reverse directions. I think the challenge is that how do you define a reverse longitudinal perturbation? I mean, in the nature, what is reverse? What is the forward path? The forward direction, that is a bit opaque to me. Yeah, I think.
One example is like in neurodegenerative diseases, it's been observed that there's a process of de-differentiation. So you have, like, let's say, neurons that are during normal development are fully differentiated into, let's say, excitatory neurons. but that with age and dementia and neurodegeneration, they'll revert back to less differentiated versions.
Yeah.
So the process of de-differentiation, and that in this clinical case, but it's also something you could induce in a lab. That sounds like something that kind of follows the path of reverse perturbation. It's naturally occurring.
Yeah. If thinking that way, I do think that can align very well. Even the reverse and forward directions, they can align very well as well. So it's not a lot of case, but in some cases we do see, maybe overexpress of certain transcription factor giving one direction. So that usually can tell you, knocking out or knocking down the transcription factor giving you the reverse direction, right? So they are usually interconnected. And that is also the point I want to highlight here. The natural perturbation or the natural development differentiation of cells, they can be trained together with perturbation data. This is the hope, actually. This is why we have a perspective paper instead of a research study. We do wish to see, either from our own efforts or the community effort, to see ablation studies showing that combining them together can really help each other.
Yeah, right on. I have billions of other questions, but I'll stop for now. So thanks for your answers.
Yeah, I can stay here a little bit longer if needed. Yeah.
Wonderful. So with that, do we have any more questions from the audience? While we do get any more additional questions, I just have to say, the fact that you guys have a robot in the lab is so cool. Like that, I just had to make the comment. Having an active learning loop is, I don't know. I've dreamed of this multiple times before, and I just had to put it out there. That is amazing. I really want to see what comes out of that, what comes out of actually considering active interventions that are proposed by the different scientists in the lab, right? Because I think that you nailed it down. At some point you said, we can't expect a single foundation model to do exactly like everything for us. Like we still are the scientists, right? We still need to go back and design all these different experiments to answer all these questions that we want to figure out. So for me, I don't know, that was music to my ears. And actually that just made me think of something. Would you say that these controlled interventions, these to me are sort of the causal directions that we are imposing on foundation models, right? Because if you have this sort of system that you're studying and you're only interested in a given subset of it, maybe supplying those interventions does set some sort of causal direction within your own research questions. But that, of course, is to be determined by each research group, as opposed to, like, there is no real biological underlying truth, so to speak. It really does depend on what you're studying, or at least that's what I think.
Yeah, I also personally agree with that, especially I think I have a slide discussion about that here. So how is it? So here, this very small fonts here. It's because I think the perturbational space or the regulation space of all the genes are quite huge. I mean, there's correlations for certain, but they can vary in very different contexts. So it's very difficult for current model to unify all your modeling across different contexts. I just want to highlight the context. I didn't even show you up here. Like a lot of the data are collected in a context of one cell lines, two cell lines, a few cell lines, but usually you're more interested in human bodies, or at least the mouse. So that is huge gap between huge challenge for generating or generalizing your model out of distribution, right? So speaking of that, building a causal model in a very strict sense is very challenging because you need interventional data in a very large space. But building a causal model with causal data in a focus state may be more feasible. Like if I only care about what happened to my T cell or primary human T cell with this interesting subset of 1,000 genes, maybe that's a more feasible question. Yeah, that's the thing I was highlighted during the slides. Yeah, that's also why I really echo with your comments as well. So in a very focused subset is more can be more accurately scientifically evaluated. But the other side of just one comment after that is that after doing this small islands after small islands, we are accumulating the data to the bigger picture. Yeah.
Agreed on all points. That's wonderful stuff. I couldn't help but think of the perturbation layer as some sort of really high signal sparsification for the model, which I think is the way to go.
It just occurred to me that because Cesar mentioned causal model, and I had some experience with knowledge graph reasoning models. So in knowledge graph reasoning, like even if you have a super large knowledge graph, like you have a lot of nodes and edges, but I think the reasoning model would, like the capability would be constrained by the diversity of the relations, and how's the relationship between the relations on the knowledge graph. So I think there may be something similar here. So even the perturbation space is very huge, but for a single cell type, for example, T cell, the logics behind the perturbation, like the important genes that would really cause some effect, those would be relatively small, because that's what this specific type of cells they are trying to do, and there shouldn't be a lot of perturbations that should affect the cell itself. So I guess it's still doable if we focus on those meaningful perturbations only, because the real biological logic behind this, it should be simple. It shouldn't be very complex. So yeah.
Okay, so any comments, any questions, anything else to round out the discussion maybe?
Yeah, I think the problem is that this presentation generates too many questions, which is an awesome thing, but I think maybe our invited guest speaker has other commitments. Because like I said, I'd have a bunch of other questions, but I think we'll table that.
All right, so at the very least, I will end the recording there. So once again, thank you everybody for coming to the 21st edition of the Multi-omics Reading Group. That ends today's session. Thank you very much.